So it does seem that filamentation is providing some advantage in a fast settling environment. So why might that be? Well, one reason that they might provide an advantage would be if they help settle more quickly. So to figure out if filaments settle more quickly through individual than lone clusters, we used this apparatus, which was filled with water. We just used a stopwatch to measure the time it took filaments to traverse this distance. Since clusters are not visible to the naked eye, we put this apparatus on a microscope and just set up a video to record how long it took lone clusters to travel that same distance. And what we found was that filaments settle almost 8 times more quickly than individual clusters. Specifically, filaments are settling at about 0.8 centimeters per second, while individual clusters can only settle at about 0.1 centimeters per second. So this is quite a big difference in settling speed, but we wanted to translate this into a currency that's more relevant for biology, which is survival. So we wanted to ask the question, if you're in a filament, what are the chances that you'll survive to the next day's selection by being in that bottom 10% of the tube compared to your chances if you're a lone cluster? So to answer this question, we took advantage of the fact that within the same tube, you have some clusters that are incorporated within the filament, which you can see at the top, and then some clusters which are unincorporated at the bottom. So we separated those and put them both through 60-second settling selection, and then we compared the amount of fluorescence in the top 90%, which would have been discarded, to the amount in the bottom 10%, which would have survived. And we found that, in agreement with our earlier observations, lone clusters have low survival. The chances that they can make it to the bottom 10% of the tube in just 60 seconds are not good. However, cooperative filaments have much higher rates of survival. Almost 80% of them can make it to the bottom of the tube in that same amount of time. So that's a huge advantage. Clearly, there's lots of reasons why you might want to be in a filament. So to recap briefly, people who might have come in late, filaments are composed of hundreds of individual snowflake yeast clusters held together by proteins. Filaments do appear to be selected for when you select for fast settling, in that they evolve more often under those conditions. This might be because filaments settle about 8 times more quickly than individual clusters, and that higher, faster settling speed does translate into higher survival. So clearly, it's great to be part of a filament. So at this point, we might ask the question, why are these filament-producing strains remaining multicellular at all? Why not just go back to being unicellular? You can grow a lot more quickly, and you can still have the advantage of being part of this huge filament that will let you settle really fast. So to figure out what's going on here, we took a filament-producing strain and we reverted it back to unicellularity. However, due to gene conversion, we still did see some multicellular clusters in this population less than 1% of the time. So when we drew these filaments up, we saw something a little unusual. This was one of those filaments, which was that all of these blue dots you're seeing here are actually multicellular clusters. At this scale, you can't even see single cells. So it appeared that there was a lot more multicellular clusters here than we might otherwise expect. So when we dissolved this filament with that same protease I mentioned earlier, and we looked at what proportion of the biomass of this filament is held in multicellular clusters, we found that it was much higher than the amount of biomass in the regular population held in multicellular clusters. So filaments do preferentially entangle clusters over single cells, and even though multicellular clusters are very rare in the overall population, we find them in a disproportionately high frequency in the filament. So it seems that unicellular strains can't take advantage of all of the benefits of being part of a filament the way a multicellular strain can. So we next wanted to know, could another multicellular strain take advantage of the filament? If we compete 2 multicellular strains, one of which is capable of forming the filament and one of which is not, what would the results be then? So we used a filament plus strain, indicated here in red, and a filament minus strain, which is indicated in blue. And we combined those at different ratios, everything from 0 to 100% and 10% increments. And the first thing we wanted to know was, at what point will we see a filament? So what proportion of the population needs to be filament plus to make a filament? And at 24 hours, we found that that proportion was 70%. Interestingly, if you extend this experiment out to 48 hours, that proportion is only 20%. So we again separated the bulk population from the filaments alone, and we ran an analysis to compare how much filament minus do we see in the filament compared to the amount of filament minus in the bulk population to try and see if the filament minus was over or underrepresented from our expectations in either group. And what we found was that filamentation can be considered a public good. So on this axis here, what you're seeing is any negative number is an underrepresentation, so less filament minus than we would have expected, and a positive number is more filament minus than we would have expected. And when you compare the bulk population to filaments, we see essentially no difference. So these other multicellular strains that are not themselves capable of forming filaments can take advantage of the filament and be incorporated within it at equal frequency as their presence in the general population. So, to conclude, hundreds of synucleic yeast clusters cooperate to form protein-based filaments. These filaments are adaptive since they settle nearly 8 times more quickly than clusters, and they greatly increase survival. Filaments preferentially include clusters over single cells, which could be providing pressure or selection, for those filament-producing strains to remain multicellular. And finally, filamentation is a public good that can be taken advantage of by other multicellular strains that are not themselves capable of filament formation. So, the big picture idea here is, the take-home message, when we subjected a new multicellular organism to really harsh stress so harsh that individuals couldn't survive, we saw them evolve to cooperate, and they did it pretty quickly. So that's all. I would like to thank my lab, our collaborators, and my source of funding. Are there any questions? So our analysis actually wasn't that good for 48 hours. It took a really long time to figure out how to do that correctly. Good question. Because since the filaments are so large and so thick, it's really difficult to determine what percentage is filament minus versus what isn't. So the results for the 48 hours were never fully analyzed because since 24 hours is the time of selection, we decided that that was the more meaningful of the two experiments. But I would expect to see the same results.
And then my other question was this, biology and your certain test substances like advantageous, maybe correlates and real life yeast that makes sense.
So we do see strains of yeast that group together in the wild. They're called flocculent yeast. If you're interested in it, someone in my lab, Jennifer Pence, is giving a talk on that a little later today. So this could be potentially relevant for wild strains, but what we are using it to look at more is sort of the evolution of uni to multicellularity and how that could have happened and sort of what factors would drive that transition or push it backwards, back into unicellularity.
The proteins involved in forming cell clusters are presumably different from the proteins involved in forming the filament, which is the matrix. But now, when you form an actual cluster, do you get the type of cell-cell junctions that get true more advanced multicellular organisms, or are they also just
So that's a good question. The clusters are the result of a deficiency in the gene that separates mother-daughter cells. It's called ACE2. So they're just remaining attached after division. It's not necessarily a protein or anything specific that's holding them together.
They're not actually aggregating, these are just...
Yes, they're dividing. It's clonal development, right? So they're dividing in such a way that they remain together. Once they hit a certain size, and that size sort of depends on what generation they are. Later generation yeast will become larger. They'll just break apart due to stresses within the cluster. But I think the other part of your question was the nature of the proteins holding the filament together.
Yeah, I'm not asking